hello, welcome to day 2 of the revision streams. So what we're going to be doing today is revising for cell biology, because on Tuesday we have our very first mock, which is all cell biology stuff. Then, what I'll be doing is, so today we'll just be doing a general overview of a topic. Tomorrow night, we will be doing an overview of some exam questions to do with cells. Then Tuesday night, I'll be doing just a general overview of some core math stuff, maybe like half an hour max. Wednesday night, I will be doing an overview of the chemistry topics. So, things like DNA replication, protein synthesis, biological molecules, digestion. I might do a little bit of it because I'm pretty sure it won't appear. Then I'll be doing a core math overview on Thursday. So we have 5 days non-stop of revision streams. So buckle in, let's get started. I've already mapped out everything here. These are the 4 main topics. You got transport across membranes, which includes diffusion, osmosis, active transport, co-transport, facilitated diffusion. Cell biology, where we talk about prokaryotic, eukaryotic. We could talk a little bit about some other stuff there, like cell fractionation. Immune system, we might talk about viral replication here, but mainly it's the humoral response and the cell-mediated response and the non-specific response of the phagocytes. And then mitosis, which is probably the easiest topic. I was able to revise this one back in year 10 without a hassle, so I think we should probably start off with the easiest topic, then we work our way up to the more complex. So, let me go move this camera in frame. There we go. Hope that's all fine for you guys. I'm going to go open my blinds a little. Okay, I think we're ready. So, let's get started. So, cell biology, we're literally just going to start off by... here's the thing, there's so much of this topic, it's going to be impossible for me to fill it all in. So, I guess we can talk very quickly about just general cell structure with the nucleus, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the ribosomes, obviously, the mitochondria, the Golgi apparatus, just the general structure of cells. So, there are 3 main structures that you need to know a little bit more in depth. So what they are is the nucleus, the mitochondria, and then obviously this is meant to be an animal cell, but we'll draw in a chloroplast there for ease of understanding, and chloroplasts. So the structure of a nucleus, I would argue, is probably the easiest one to understand. So, I'm drawing it like this. So what we have here is the nucleus, or the nuclei. So, that right there, that little, I guess, orb, is the nucleolus. And that is where the ribosomes are actually initially synthesized. Then we have obviously the chromosomes, or I'll just put DNA for ease of understanding. You have obviously the nuclear envelope or the nuclear membrane. I like to call it the nuclear membrane, but you can call it whatever. The nuclear pores, the holes which actually allow things to enter or exit the nucleus, I guess allow chromosomes to enter and exit. And then right here, the nucleoplasm. So that's the structure of a nucleus. Very easy to understand. Yeah, I would argue that's incredibly easy to understand. Probably the most simple one. Then we move on to something a little more complex, which is the structure of a mitochondria. Now this one has a matrix. It has plasmids, which is circular DNA. It has small 70S ribosomes. And little thing you should always mention, the 70S or 80S ribosomes. 70S obviously kind of proves a theory of evolution because it suggests that these cells were kind of the precursors, that they kind of coexisted in a way. It's an interesting theory. So I am thinking, what else is there here? I guess you could talk about the matrix, and there is one word which I've forgotten. Is it cystine, or is that the other one? The folds on the membrane. I believe those are called cystine, but I could be wrong there. So I'm going to leave that one as is, and we'll come back to it in a bit. Again, what I like to do is I like to just dump everything that I know initially without looking at any books, anything, just all off the top of my head, even if it might be slightly inaccurate. That way it at least allows me to correct my mistakes. And then finally is the structure of the chloroplast. So you got your thylakoids and grana. Hold on. Which way is it? Thylakoid is the individual, disc. Grana is the plural. I hope to God that's right. Then you have the actual substance, which I don't recall what it is. Obviously, this is, I forgot to kind of mention, it's a double membrane. It also has 70S ribosomes. And obviously, it also has circular DNA. So, the thylakoids contain the pigment chlorophyll. Which is a photosensitive pigment. So it basically absorbs the light, using it for photosynthesis. I don't recall, and this is really going to tick me off now, so apologies there, but I didn't get it. But there is two words I'm missing. One word for the actual substance here. I don't recall what that is. And I don't recall what the little things connecting the discs are. So what I'm going to do is I'm going to draw arrows out to them. And I'm going to hope that later I can kind of recall. So there we go. Those are the three main structures right there. The nucleus, mitochondria, and the chloroplast. Let's go take it back now, and let's just talk about some general stuff. I'm going to have to crouch down now. You got the rough endoplasmic reticulum, which, in summary, is kind of like a set of maze-like structures. That's how you can identify it, with ribosomes that are kind of like embedded into it. I forget the term for embedded. I know there's a word for it, but basically, this just synthesizes proteins. And like a lot of things in cell biology, refer to a large surface area, like the matrix has a large surface area, rough endoplasmic reticulum has a large surface area. The smooth endoplasmic reticulum is a set of tubular... Oh wait, hold on, these are folded membranes. The smooth endoplasmic reticulum is like a set of tubular membranes which synthesize lipids and carbs. Now, the point at which these kind of join is the Golgi apparatus. With the Golgi vesicles. So the Golgi checks and modifies proteins, but we can be way more specific. And the vesicles, I've already explained so many times what these do, so I won't write too much. But basically, it's used in exocytosis, which is where it basically fuses with the cell membrane to release its, I guess, whatever's in it. It releases its contents outside of the cell. And that's called exocytosis. So it checks and modifies proteins, but we can be way more specific here. So what we can actually say here about the Golgi is that it packages proteins, it creates kind of glycoproteins, glycolipids. In other words, what happens is after it's synthesized here in the rough endoplasmic reticulum, it goes into the Golgi. The Golgi then obviously checks the protein. It attaches other components to it, such as carbohydrates or lipids. It then packages the, protein within a vesicle. This vesicle then fuses with a cell membrane to release the, its contents via exocytosis. There is a 4-mark question about what the Golgi does, and I got 3 out of 4 marks for it, so I missed a point. So that's why I think you guys should revise the Golgi. Because that summary there that I just gave was not enough. So you gotta take a little bit more detail. What I'm gonna do, this might take a minute, but I'm gonna go rub out all of the names. So please bear with me here. Nucleus. And that's it. So, I feel like we're missing something. Am I being silly? Are we missing something? I guess we could talk about the cytoskeletal structure, which is basically just like the... I don't even remember what that is. I don't know. Ultrastructure is a term that can define kind of, specifically what organelles are in a cell and like kind of why. I am thinking now, I don't think that there's anything I've missed. So that's all stuff there. This whole little corner is all of the cells and the organelles. But we've missed out some key points here. We've missed out the difference between prokaryotes and eukaryotes. So let's very quickly draw a table. I have already done this. in the previous revision stream. So let's see what I can remember. So eukaryotes obviously have linear DNA. Prokaryotes' DNA is circular. Eukaryotes are attached via, well, they are wound around histones, which are basically just support proteins. Histones do not exist in prokaryotes. Eukaryotic DNA is longer. Prokaryote is shorter. There were a couple more words here, but I've forgotten. Prokaryote is supercoiled. Yes, that's a word. It's supercoiled. So basically, it's supercoiled into the prokaryotic structure, and it's attached to the membrane. Meanwhile, in eukaryotic, it exists as chromosomes, which are in the nuclei. So there you go. Very, very brief overview there of the difference between eukaryotic and prokaryotic DNA. We'll draw like a little arrow there to talk about that. So we have a lot more to talk about. Should we do, again, I'm just thinking of stuff as it's coming to my head here. So it's very probable that I miss some stuff. This is all that I'm remembering. Should we quickly do homogenization, centrifugation? Okay, so homogenization. So this is part of the cell fractionation topic. So the way this works, in summary, is you get the tissues from, I guess, the cell sample. You kind of homogenize it. So you get a homogenate. You filter for any debris. So you've got your homogeneous solution, and then from there, you would use a centrifugation, a centrifuge, and spin it at different temperatures. Jesus Christ, what am I on about? Different speeds. And what this does is, for more dense organelles, will form rich pellets at the bottom, and obviously the lighter, less dense organelles will require a higher speed in order to form a pellet. Now, in order to do this process, you're going to need to use an ice-cold buffer solution. So a reason it's ice-cold buffer solution, ice-cold obviously slows enzyme activity. So no destructive enzymes will be used. Buffer keeps the pH neutral. This is so that obviously it doesn't kind of damage anything. And then the solution so that there is no net osmosis, I believe. That is the first thing that comes to mind. Although, why would there be osmosis? And I might need to come back to that. Yeah, I was going to say, why would there be osmosis and self-fractionation anyways? That seems weird. Okay, I'm back. So that's self-fractionation. We're going to need to take a little bit of time when we're done here to go back and look at just the structure of the chloroplast and then obviously self-fractionation. But I'm happy with this so far. We've done a lot here. In a short amount of time, we've covered a lot of organelles. We've covered the difference between eukaryotic and prokaryotic. Should we talk about I don't know. I'm thinking now. What other topics are there to talk about before we get onto things like mitosis? Should we do mitosis now? Get it over and done with. So this one I love because I made an acronym for it. IPMA TC. So I prepare meals at the college. I came up with this one 2 years ago, and it still works. You got interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. So mitosis in itself is genuinely one of the nicest topics, but you can easily get mixed up a lot by some of the things. So firstly, interphase, it refers to three things. Obviously, it's regular cell activity, but you got G1, G2, and I think it's R. So during growth 1, that is when your organelles replicate. Then you got DNA replication. And then in G2, everything is checked to ensure things like cancer don't occur. In other words, an uncontrolled cell division. In other words, making sure that nothing went wrong with the division, which can be flawed. That can be a flawed thing. So I'm just going to put there G1, G2. I'm going to have to rewrite it. Because I kind of messed up there, put it in the wrong place. So that's interphase, which is, regular cell activity. Because you have to remember that whole little pie chart thing that the teachers love using to point out the the little amount of time that mitosis is actually being present. And yeah, it's true. Obviously, mitosis only takes up a small amount of time. So, prophase is when shit starts to get real. That's when the DNA condenses. Or I guess the chromatids. The chromatids condense, so they become visible. And I believe this is when the nuclear membrane kind of breaks apart. Oh, that might be the next bit. Shit. See, I don't remember much here. That's when the nuclear membrane, I guess, evaporates. So, fuck, you know what? I'm gonna have to. Because I know this topic so well, but I forget what happens in each individual stage. If you get an exam question about, describe mitosis, you probably don't need to include the names of the phases, and so I probably do really well. But if it asks me specifically what happens on each phase, I'm screwed, because I don't recall. So let's just go spend a minute to check. Obviously microscopes too, yes. She did actually she's repeated multiple times about microscopes. So you know what, we'll take a minute to do it. What was I meant to be looking at here? Ice cold buffer solution. pH stays the same, does not denature and change enzyme activity. Oh yeah, no. Isotonic. Isotonic cold buffer solution. Yes, I was right. I was absolutely correct there. Right. So let's have a look. So in interphase, you got growth 1, synthesis, and growth 2. Made a little typo there, but that is what it is. So in prophase, the nuclear membrane breaks down. Yes, I knew it was in prophase. You fuckers. So that's when it breaks down. The chromatids appear condensed. Yes, it was chromatids. And the spindle fibers begin to form. I guess we can just put spindle fibers form. Then the chromatids coil and line up at the cell's equator. Centrioles send out spindle fibers, which are now at opposite poles. So okay, then anaphase is where the chromatids are pulled apart. And and a V-shaped appearance. Then the telophase is where the cytoplasm divides. The cytoplasm divides as the chromosomes are now, so obviously two. And then cytokinesis is when the membrane forms to create two. Okay, cool. That is actually quite a lot of information. So let me close my eyes. Let me try and recount it from memory before obviously cheating. So interphase is where you got G1, G2, and DNA replication. Prophase is where the nuclear membrane breaks down and the chromatids appear condensed. I just punched my hand. Ow. I cut my hand by the way. So IPM. So metaphase is where they line up at the equator and they coil. Is it called? Yeah. So the chromosomes coil, they line up at the equator, and the centrioles line up at the opposite poles and they send out the spindle fibers. Anaphase is when the spindle fibers attach to the centromere. Centromere? Yes, they attach to the centromere, so they would attach to, I guess, a middle bit, pull them apart, and this forms a V-shaped chromosome. Now the chromatids are in metaphase, the nuclear envelope reforms around the chromatids, and the cytoplasm begins to divide. And then finally, in cytokinesis, you have two identical daughter cells, and the nuclear membrane, the cell membrane reforms. So it honestly, it's not a difficult topic. It's not actually a bad topic. I'm going to be completely honest here, that's not a bad topic. You literally just talk about the chromosomes condensing, lining up at the equator, becoming V-shaped, and that's it. Spindle fibers, they form, they get sent out, they attach to the centromeres, and pull apart. The cytoplasm divides, the nuclear membrane breaks down, the nuclear envelope rebuilds. It's honestly easy. The only thing I would say would be difficult is just remembering a couple key words. So those key words would be spindle fibers, centromeres, equator, poles, chromosomes, V-shape, cytoplasm, daughter cells. So you can also use this, there's kind of a thing here, anaphase. So if you're trying to identify anaphase, obviously you look there, V-shaped chromosomes, that's what we're looking for. Prophase, we're looking for obviously the condensed chromosomes, and you see the nuclear membrane begins to break down. The metaphase, you can see that the chromosomes are lined up at the equator, and cytokinesis, because there's two identical cells, and the nuclear envelope is reforming. But mainly the divided cytoplasm. So, let's go talk about the phospholipid bilayer, because I can't be fucking bothered to continue rambling about mitosis. So, you got the polar head, basically, of a so it's hydrophilic, and the non-polar hydrophobic. So one of these is saturated, one of these is unsaturated. You got your phosphate group here, your fatty acids, and your glycerol backbone. These are obviously attached together via ester bonds. And these, obviously, because of the way that it's hydrophilic and hydrophobic, the hydrophilic head will face the extracellular liquid, basically, and the hydrophobic will point away. It will kind of face inwards. Meanwhile, the hydrophilic will face outwards. This creates a phospholipid bilayer because in order to remain hydrophobic, in order to stay away from the liquid, or in order to stay away from the water, there has to be two hydrophobics. So yeah, phospholipids form a bilayer because the heads will face the aqueous solution, the tails will move from the aqueous solution. So phospholipids form a layer so that the tails keep facing inwards. And the phosphate groups will interact with the thing. So here, this will obviously face away. So it forms a bilayer. It forms a bilayer because, again, the phospholipids form a layer, so the tails face inwards, the phosphate group faces outwards. So, there's a lot to talk about, actually, with this topic. Should we firstly just talk about the different types of movement across cell membranes? Honestly, for this you only need to know a handful of definitions. So you got, obviously, simple diffusion, which is the movement from an area of high to low concentration. So basically it's going down the concentration gradient. And that's all you need to put. Simple diffusion is the movement of molecules down a concentration gradient. I got you, it's not. You need to also talk about the fact that this is only for non-polar, small, and lipid soluble. So there you go. So, simple diffusion is the movement of non-polar, small, lipid-soluble molecules down a concentration gradient. Boom, 2 marks, easy. Facilitated diffusion is a tad more complex. I'm taking the piss, it's not. It's literally identical to this, except you get rid of the non-polar small part, and instead you put it requires a transport protein. You should also mention that this is a passive process. So, there you go. Next is osmosis, which is literally identical to simple diffusion, same old definition, except this time you put down a water potential gradient. We will actually go back to osmosis in a tad. I actually made a song about osmosis featuring osmosis T. You got active transport and co-transport. So active and co, both exactly the same. They go against the concentration gradient. They always involve the use of a transport protein. Co-transport requires... Okay, I've drawn a blank here, because I know there was a beautiful mark scheme where all 5 of these terms were defined perfectly. And I can't recall it now. But active is a active process, so it requires uses ATP to travel against the concentration gradients using a transport protein. That would be my definition of active transport. Co-transport is, again, an active process going against a concentration gradient using a transport protein, but it involves the movement of 2 substances. The movement of 2 substances against a concentration gradient using energy, or using ATP. I'm going to have to go grab my computer because I'm real... In fact, you know what? We'll draw a blank. Good thing that we remembered that, because there is a really nice mark scheme which genuinely, it sums up this topic so well. Anyways, we have not done everything yet. We need to still talk about the structure of the membrane. So you got things like, and I'm drawing a blank on the name here, cholesterol. You got things like your glycoproteins such glycolipids, and your obviously channel and transport proteins. So cholesterol controls, I guess, the rigidity of the cell membrane itself. I guess how... Oh, and I'm gonna write that for later as well. I see. I'm drawing a blank now on cholesterol. Cholesterol controls the flexibility, so it controls the flexibility. Glycoproteins and lipids are recognition sites, or basically receptors. And I think that's everything. So yeah, cell recognition. Also for enzyme signaling. Transport proteins, you should mention that they are is it philic? Yes, hydrophilic. So that's obviously, are they intrinsic? Yeah, intrinsic because they go all the way through. And they allow molecules to enter or exit the cell. Now, the thing you should also mention is that the membrane is selectively permeable. Now, the fluid mosaic, I always draw a blank on why it's called this. I always draw a blank on why it is specifically called fluid mosaic. If any of you guys know, now will be the time to say it, but fluid mosaic, I'm going to say fluid because it contains a variety of components, different components, and those components have the ability to move. You know what, I might actually have my past paper here. Let's just very quickly go through, make sure. So yeah, osmosis. So yeah, it's from a dilute to a concentrated substance. Oh, here, hold on. Okay, yes. There was more to osmosis. I knew it. Hyper, iso, hyper. Hyper is when the solute is lower, so it's a dilute solution. Hypertonic is when the solute is higher, so it's more concentrated. I forget what that exactly refers to. Crenation is when the cell shrinks. Lysis is when the cell bursts. Isotonic is when there's an equal concentration. So hypertonic is when there is a lower water potential outside the cell, which means that it's going to obviously shrink because water is leaving the cell. There you go. So we got the cell and water leaves to the outside. Hyper is when water enters. There you go. So all you need to do to describe osmosis is talk about low water potential, high water potential, dilute, concentrated. So for example, we have a hypertonic solution. So that means that there is a higher concentration of water inside the cell, which means that it's a concentrated solution outside of the cell, which means, it's a concentrated solution outside of the cell. There's more water inside of the cell, which means the water inside the cell will travel via osmosis out of the cell, which will cause crenation. There is also some words here. You got turgid, plasmolysis, and incipient plasmolysis. So turgid is when the cell swells and the protoplast pushes against a wall. These are some tough definitions for some reasons. So I'm gonna maybe not write these down, but I'll just remember them. Turgid, plasmolysis, and incipient. So yeah, hypertonic is when the higher concentration, so it shrinks. Hypotonic, just think about it this way, hypotonic because it explodes, hypertonic because it gets smaller. Hyper, smaller. Anyways, active and co-transport. So, oh, fuck, you know what I've forgotten about? The sodium-potassium pump. Are we going to need to remember that for this exam? I'm hoping we don't, but let me recap this. Fuck off. So yeah, active transport is an active process which requires energy from ATP. A process is in which molecules move from an area of low to high concentration against a concentration gradient involving transport proteins. So, the way that the sodium-potassium pump works is that sodium, first and foremost, binds with the pump. Phosphorylation via ATP occurs, which causes the protein to change shape, releasing the sodium, making potassium bind to the protein, which releases a phosphate. So, in other words, how are we going to draw this? Okay. I think I got it. So, here we got sodium. Sodium binds with the pump, but it can't get through. So what ends up happening is ATP will, I guess, release an inorganic phosphate molecule, which will bind to the, what is it called? See, there's no real way of summarizing the NK pump. It's really annoying. Sodium, so the pump has an affinity to sodium. So sodium binds with the pump. ATP phosphorylizes the pump, which causes the protein to change shape. This releases the sodium to the outside of the cell, and and now it has an affinity for potassium. Potassium causes the phosphate to release. Like, I understand it, but it's a pain in the ass to explain. And finally, I don't even really want to even refer to co-transport because it's so annoying. It is so annoying. I guess you could quickly refer to symporters. Yeah, honestly, it's such an irritating topic here, co-transport. So, basically, sodium ions transfer, I'm doing this from memory, sodium ions go out of the cell, out of the epithelial cell, via facilitated diffusion. This then, hold up. Sodium leaves via facilitated diffusion, leaves the cell via facilitated diffusion. This means that the cell now has a low concentration of sodium. This means that sodium from the lining, the lumen, will diffuse in via facilitated diffusion, but they will also bring with it glucose. That's it, right? Number one, yeah. So sodium ions are transported out of a pump Sodium ions are transported out of the cell and into the bloodstream. Oh, it's via active transport. Okay, I thought it was facilitated diffusion. But no, because it's a sodium-potassium pump. Okay. Sodium ions... Okay, hold on. This means there is now a low concentration of sodium ions in the cell compared to the lining. Sodium ions enter the cell via facilitated diffusion using a symporter. That was a word I forgot, symporter. Sodium passes through the transport protein and it brings glucose through. The concentration of glucose and sodium is now higher in the cell than the blood. So glucose travels out via facilitated diffusion. Sodium ions move out via the sodium-potassium pump. Okay, hold on. So we almost had it there. So it's the NAK pump, firstly. This creates a low concentration of sodium. So this means Na moves via facil diffusion, whilst also using a symporter. Glucose is brought through. There is now a low conc of Na plus So, this looks like some madman writing, but The sodium potassium pump is used so that sodium sodium moves out of the epithelial cell. This creates a low concentration of sodium in the epithelial cell. So sodium moves via facilitated diffusion in a symporter, whilst also bringing glucose with it into the epithelial cell. This now creates a low concentration of sodium and glucose in the bloodstream. So then glucose moves via facilitated diffusion out of the bloodstream, and sodium obviously moves via the sodium potassium pump. I am getting real tired now, so let's wrap things up for today by doing just one or two more things. Firstly, the immune response. Let's get this over and done with, because it's rather quite easy. So we have phagocytosis, in which a pathogen is surrounded in a vesicle. Well, it's engulfed and trapped in a vesicle. It then joins with the lysosome to form a phagosome. This then releases via endocytosis hydrolytic enzymes. And so this breaks down and digests pathogen. From there, the phagocyte then presents the antigens. It presents the antigens. This causes the specific helper T cell to, I guess, bind with it. And from there, the helper T cell is activated. So the helper T cell binds with the complementary. The complementary helper T cell binds with its antigen, which activates it and causes it to divide by mitosis. From there, the helper T cells will trigger a humoral or a cell-mediated response. Cell-mediated response is when they activate cytotoxic, which release perforin, which is an enzyme which perforates the cell, causing it to lyse. Meanwhile, the humoral response is where you got your B cells, which divide into memory or plasma. Plasma B cells then release antibodies. Memory cells remember a complementary antibody and antigen combination. Antibodies will either agglutinate or neutralize. So agglutination is where it basically just sticks a bunch of the cells together. Like so. Neutralize is where it just completely prevents the attachment of the cell. That is my rough example. I might have missed a bit. The most important thing is the helper T cell binds with the presenting antigen, which activates it, causing it to divide, and then from there you know the rest. So, before we wrap up, I want to go quickly to the fluid mosaic, which I don't recall where I could have possibly put this. Oh, hold on. A little bit about antigens here. Sorry, antibodies. The structure of an antibody is, this is the variable region. This is the binding site. This is the structure that says the same. And this is the heavy chain. This is the light chain. But I think that's about it. So let me go quickly find, if I still have it, Okay, hold on. Let's just check we've done everything. We did not do microscopes, but everything else we've done. So, microscopy and methods of studying cells. I guess that would be fractionation. So next time, we're going to do microscopes. That'll be tomorrow. And we will also go quickly revise, I guess, more transport across membranes. We can revise that a little bit more. But I think that's honestly a good place to stop. We've spent an hour. We've gone through a lot of stuff here, so this has been really good. Really good. I just want to check quickly. And maybe we review mitosis just a tad more? Yes, this right here. This was what I was looking for. So cytoplasmic sodium. So sodium in the cell binds to the NAK pump. The binding of sodium stimulates the phosphorylation by ATP. Phosphorylation causes a protein to change its shape, which releases sodium. Then it now has an affinity to potassium. So it binds to the potassium binds to the carrier protein, which triggers a release of a phosphate group. Okay, yeah, I remember that now. Just trying to find it. There were some really weird questions actually to do with this. But I don't seem to have it. So next time, so tomorrow, what I'm gonna do, next stream, we will revise microscopes, a little bit of cell membranes. Mainly, we might have a look back at osmosis and co-transport, active transport. We will go and quickly revise mitosis. So microscopes, membranes, mitosis. Those are the three things that we should revise. I feel like tomorrow I don't even have biology, which sucks. So I'm going to have to do this revision in my own time. But thank you guys for watching. As always. We have done a lot today. Christ. Now I'm going to chill for a bit, and then I think maybe later I'm going to do some of these quiz things. Basically, it's this website which is there's a bunch of biology-related quizzes. Thing is, it doesn't particularly help me with learning. It more just helps me very quickly remember some key points. But they are kind of forgettable, so I might not do as many. In fact, should we do some now? If you guys give me a minute and a bit, I'll go do some now. I'll go grab my computer. Hello, I'm back. So, let's go on this website just for 5 minutes and we'll go do some quick revision on, should we do membranes? We'll just revise the membrane topic. Because there's some quizzes here with like 90 questions. I'm not doing all of them today, guys. So, Okay. Cell recognition. We'll do one on mitosis right now. 20 questions. Okay. What's the correct order? PMAT. Prepare meals of a college. During which cell cycle stage does DNA replication occur? Interphase. During which phase are the chromosomes aligned along the equator? It's not prophase, it's metaphase. Fuck off, Apple. Which phase do the chromosomes condense? That's prophase. What part of a plant would be a good place? The root tips, because that's the only place where the plant can actually divide by mitosis. Via meristem cells, I believe. Which cells are not produced by mitosis? Gametes. Yeah. Chromosomes are not visible in what stage of mitosis? I guess interphase. Spindle fibers connect centrioles in this stage. That would be anaphase. Numerous mitochondria are needed around the spindle fibers. I don't know what stage that would be. Maybe anaphase? Where they're being released. Spindle fibers disperse. I guess after anaphase, so telophase. DNA is replicated, interphase. Nucleoli disappear and the nuclear envelope in prophase. Spindle fibers are made from microtubules, I believe. In most cases, mitosis follows it, so it's two daughter cells with an exact copy of DNA. Why might the genetic via mutation? Chromatids are one half of a chromosome containing an exact copy of DNA in the other half, I think. The name described a point on a chromosome is a centromere. Once the replicated DNA molecules are separated, they are referred to as chromatids. What happens to spindle fibers during telophase? They disintegrate. What is the name of the organelle that moves centromeres? Centrioles are not present in what are the following? Plant cells. Cytokinesis follows what stage? Telophase. Okay, I hope I did okay. %, cool. Spindle fibers connect during metaphase. Okay. I might have... I mean, no, I guess the spindle fibers are sent out and they connect to the centrioles. Anaphase, yes, okay, no, that makes complete sense. That was a bit of a mistake. They are referred to as chromosomes when pulled apart. Damn it. I got this the wrong way around then, because I called them chromosomes after the fact. It's not. Okay, I really just want to quickly do one on cell membranes. But I can't find it. I literally cannot find the questions on... Hold on, guys. You're going to have to bear with me. Weird. Okay. Should we just quickly do electron microscope quiz? So, the following microscope uses electrons to produce, that would be a scanning. A 2D image would be a transmission, because they go right through. A limitation of electron microscopes is that not a limitation. The transmission electron microscope can usually magnify, I don't know, like a 10K. The scanning electron microscope, I don't know. I have no idea. I'm going to put 30K. The electron beam has a wavelength that is longer than that of visible light. Sorry, it's shorter than visible light. Electrons used in a microscope are, will they be negatively charged? I don't know, actually. What will they be? I'll put negative. Electromagnets can be used to focus a beam. The fact that they have a positive charge. Modern electron microscopes can resolve objects that are 0.1 micrometer. I'm going to put. Why do electron beams need to be in a vacuum? Because electrons can be absorbed or deflected by air molecules. The electron microscope transmission it passes through a thin section. The beam is directed onto the surface. Oh, here, hold on. Nah, it's fine. Which electron microscopy technique can be used to generate a 3D image at scanning? Which of the following could not be resolved with an electron microscope? Probably, I don't know, an individual protein, because that's like a molecular level shit. Which of the following can be imaged using an electron microscope? Dead specimens. Well, what is the term used to describe an image derived? Probably a micrograph. Why might details be open to doubt? Electrons might have altered the structure. It's either this one or that one. What is an artifact? An unnatural structure seen that results in from the sample preparation. Why might the 0.1 nanometer resolution in the transmission electron not always be achieved? Artifacts might lower the resolution, maybe? What problems can be caused by the high energy beam? Specimen could be destroyed. Honestly, I'm a bit shaky with that topic, so let's see. Probably didn't do that well. 75%. Okay, cool. This was a tricky topic. Transmission electron is that 100,000? 100,000. Scanning is 30K. Wow, okay, that's a big difference. The electrons have a positive charge. That's cool. You can see 0.1 nanometers, that's cool. Scanning, obviously, wait, no, hold on. TEM and SEM can see 3D images, that's cool, I didn't know that. Artifacts are unusual structures that result in sample preparation. Okay, cool. So we did alright there, considering we haven't really covered that topic. I forget what magnification and resolution mean. What is the resolution limited by? The wavelength of light and the distance between two objects. I think the resolution is just how clear it is, the difference between two images. The magnification is how much an image has been increased by. Beyond the limit of... In fact, hold on. What are they? Because I always fucking forget. Resolution. So magnification... How many times the original image is magnified by? And resolution is... Magnification is how many times the image has been magnified by. Resolution is how, I guess, how clear an image is. Huh, this is weird. Some early microscopists claim to see a tiny man in nuclei and sperm cells. Apart from their bias and imagination, what might have led to these observations? I guess artifacts? What type of radiation is used in a compound? Light. An object being imaged is usually mounted on a stage. The limitation of a light microscope is it can't see detailed structures. A benefit of a light microscope is that it can image living samples. The ability to distinguish 2 points on an image, there you go, resolution. How many times bigger an image is compared to the object, that's magnification. Optical microscopes are convex or concave, I forget the difference. The wavelength of light is, I think, 0.2 UM. How many lenses are present in a compound microscope? I think 2. Millimeter, nanometer, so it's nanometer, micrometer, so 2, 3, 1, 4. What the following could not be resolved? An individual ribosome. You could probably see a bacteria. 84%. Didn't know that. It's usually mounted on a slide. Oh yeah, no shit. For wavelength, there is no further increase in resolution. Okay. Well, we did alright considering most of those topics I haven't revised in literal months. So, moral of the story, guys, revise both topics. Revise both topics. I forgot what I was going to say. Revise both topics more, don't just revise the chemistry topic, because I do that and I kind of fucked up as a result. Now I have all of this shit to revise in under a day. Thanks for watching.